All right, so the question was asked, how did we end up with mRNA vaccines over all of these other options? And we're going to work our way toward that conclusion today. So, vaccines are super popular uh, medical remedy for viral infections, probably because they gained extraordinary fame with a few, really just a couple of viruses that they absolutely crushed out of existence, most notably smallpox, uh, polio, measles, a few others that were really pernicious diseases that most of the world was suffering through kind of consistently. The data doesn't go that far back, obviously, for things like polio, but uh, so there was a lot of hope that the vaccines in general would solve all of our problems. Unfortunately, as you well know, even before COVID came along, the vaccines were particularly challenged by respiratory viruses, the flu being the notable example, such that you could put a dent in flu cases, but it really relied on people getting a new vaccine that re-educated their immune system every year. Um, and again, my favorite word, the virus was a moving target. It was changing, constantly mutating. You're always playing catch up, trying to make a new vaccine that'll be able to target it. So, in addition to all of the treatment modalities we talked about in the last lecture, we have the pursuit of the ideal vaccine in COVID, and we're going to talk about all the different options that were investigated starting now. And I think that the best way to think about how to arrive at the best vaccine starts off with asking, what is your body, or at least reminding ourselves, what do you normally do to fight back against COVID? What are the immune pathways that lead to the kind of memory defense that you hope to incite with a vaccine?
Mm-hmm. I think that's a good. I think it's a good introduction.
So, the vaccine is ideally supposed to trick your body, if I can say it that way, into believing that there's something it worth mounting an immune response against. However, ideally, that something isn't going to mount nearly as crazy of an immune response as you would have to the normal particle, say the normal virion, and it wouldn't be uh, obviously since it's not an infectious agent. Hopefully, it won't be destroying your tissue and causing all of the fun, not so fun pathologies that we've been studying up to this point. That also seems to be a huge challenge, though, because half of this disease, at least COVID-19, seems to be your body's reaction to the virions themselves. Uh, a lot of the severe disease is coming down to these thrombic events, coming down to macrophage activation syndrome, i.e., cytokine storms. And so it's not entirely clear how to tell your body that there's something worth reacting to, but don't react too much. And so that's been a huge part of the challenge. And we'll see how that sort of led to the weeding out of some of the vaccine candidates.
Mm-hmm.
Um, so I think without complete spoiler alert, the teeter totter is always going to swing between efficacy and safety. Some of the vaccines may prove more effective than the one we ended up with, but they're not considered as safe. And we'll get into the details of why those determinations were made. So I pulled this figure out of a Nature article, which I wish we would have had time to read too. This is just looking at how the vaccine was developed over the last year or two. And what they're trying to remind us about here is that this is normally how your body, this is essentially the basic idea of how your body is going about immediately attacking the viral process, if I can say it that way, attacking the virions. through these two T cell mediated processes, essentially. The T cells are able to actually attack the various proteins of the virus independently that are able to lead to.
Use the cursor so people can
right.
see what you're pointing at. Yeah, there's the laser point.
So these were some of the more acute, immediate strategies that your own body had for reacting to the presence of these epitopes. And so the thought was that if you could do what the T cells are doing, if you could incite the T cells to perform this way, that's really more like it. If you could get the T cells to do their job without an actual infection being there, and you could get these processes to happen, the attack of these NSPs, then you might spike proteins. If you could get them to actually go after the virions, you would be able to achieve your ends without sacrificing any of your own tissue.
What's the difference between the left and the right side of the graph here?
I'm more interested in the right side. I'm not sure.
And then
The left side is actually just trying to tell you that there's these processes that lead to uh, this is the virus's defense system essentially, but you're also like normally your body's gonna have this transcription. We've talked a lot about NF kappa B being the first transcriptional activation of multiple inflammatory cytokines. And so one of the things the virus does is interrupts this. It can also interrupt this interferon process. This low interferon is what they're meaning here. The left side's kind of a terrible drawing. That's why I didn't want to get into it. But these would also be vaccine strategies would be to somehow attenuate this, stop the virus from doing these processes that it's doing.
And on the right side, the CD4 cells and the CD8 cells, they're shown to be targeting different proteins.
Mm-hmm.
And then, so I was just thinking before we started this slide that I wanted to add something about B cells.
So, yeah, they don't even mention B cells here, but obviously B cells are your plasma cells. They're the ones that are going to be making the antibodies ultimately. The T cells here, this isn't shown unfortunately, but the CD4 T cells are going to be the ones that are instructing the B cells on how to make those antibodies inside of the lymph nodes and spleen. So those are really important. These CD8 cells.
What's the difference between CD4 and CD8?
Right. So CD8 cells are known as killer or cytotoxic T cells. CD4 cells are helper cells. They're both called T cells because they originate in the thymus, but they have pretty different jobs within the body. And the CD4s, they have a lot of jobs, particularly a huge part of their work is grabbing a hold of these different antigens and teaching the B cells to make antibodies against them. They can also grab a hold of these antigens and teach and set off different cascades to ramp up inflammation in the body in general. The CD8 cells actually go and attack infected cells. That's the main job of these guys. They walk around your body and they knock on the doors of all your cells and make sure that there's no bad guys living in there, basically.
So would you say that the CD4 cells encounter, like, what is the difference in the way that these two cell types encounter antigen? Because the CD8 cells are killer cells. Are both of them encountering through the MHC? Or can a CD4 T cell actually interact directly with a virion?
Yeah. Like, can it chew it up and process it? I think that they can do antigen presentation also, so they would be like eating up little pieces of it and presenting it as well. Yeah.
But all of this is centered on this idea of antigen presentation. So it's showing that the CD4 T cells, the CD8 T cells are able to react to all of these antigens, but it's not like they're reacting to the antigens just floating around in space or just floating around through interstitial fluid. All of this requires antigen to be presented on the surface of antigen-presenting cells. So we talked a while back about the MHC, which is the major histocompatibility complex.
I guess, yeah. Sorry to interrupt you. I think that more often we have the CD4 cells are receiving presentation from other people in the body, and they're going off and doing their job to activate further cascades. More because the cells that are doing a lot of the presenting are the ones that eat stuff. T cells aren't usually eaters. The macrophages, uh, other like the monocytes, the immune cells that are going around gobbling up pieces of virus and presenting it, or infected cells would present it also. But the helper cells, once they have the antigen presented to them, are able to create cascades that target these different structures.
And what you can see is that the virion, which is drawn here, when it's present, it's said to lower the presence of MHC class 1 molecules, which is basically like putting on an invisibility cloak, because these MHC proteins are antigen-presenting proteins. They carry around chunks of antigen that these T cells are able to sense. And when they sense them, do the T cells take the antigen away to go present it to a B cell? So basically, the T cells, when they first encounter an antigen that is being held out by, let's say, a lung epithelial cell, because all of your cells have these MHC compounds on them, MHC proteins, it actually takes that antigen and then carries it away to be able to bring it to B cells and to tell the B cell that, hey, this is the antigen that you need to be training for because I found it in the body already, and you need to be becoming more and more mature.
And dendritic cells, other cells in the body can do the same thing. They'll migrate to the lymph nodes where they can interact with B cells.
Exactly.
But the T cell helper cells, also the CD4s, they can do more than just educate B cells, but that's a huge part of their job. They can also release inflammatory cytokines to ramp up. If they find the site of the infection, for instance, like in the throat or the lungs, they can recruit more help to that area.
And the idea of the vaccine is that, the vaccine is able to get into the middle of this and not have the actual virion there that's capable of replicating, but having the virion that's there that's able to allow B cells to start to produce antibodies by way of these T cells. Because if your cells start to produce spike protein, and that spike protein is then chewed up by an antigen-presenting cell, it's going to start showing pieces of spike protein on the surface. Your T cells are able to take that to your B cells, and your B cells will produce antibodies to basically all epitopes that are on that spike protein. And that's how you have an effective response without ever having to go into the whole viral cascade.
So exactly. So these are all potential targets, essentially, for the, these are all potential antibody targets.
And our ability to have vaccines has been a long road. So before we kind of get into the technologies and answer...
One more thing to add.
Oh, please.
So in addition, we're going to be talking a lot about antibodies, right? Vaccines ramp up your antibody production. Another strategy that we're going to encounter is people are trying to design epitopes, especially on some of these multiple, what do they call them? Multiple subunit?
Just
subunit type vaccines.
Yeah.
Is they're looking for epitopes that bind T cell receptors as well. So it's not just antibodies. Okay, that's all I want to add. We'll get to that though.
So to talk a little bit about the arc of vaccines and issues with our old technologies of vaccines and new technologies of vaccines, we have to kind of go all the way back to smallpox. Because smallpox was the first illness that people developed any kind of vaccine for. And we're talking late 1700s, where they started to realize in the West, because obviously this was already known in China, this was known in India, I think this was known in Africa. It filtered to Europe in the late 1700s, and Europe is basically like, oh, well, we invented it. But the principle was that people realized that those who had experienced smallpox, which was a devastating disease, I don't have any pictures of it in the presentation because it's horrific.
Not safe for work.
Not safe for work, not safe for life. I mean, I think that it's worth sort of looking to see at it, but I didn't want to spring it on you guys. But basically, people had come to realize that there were certain people who were immune to reinfection. If you managed to survive smallpox, you didn't seem to get it again. And so it was a pretty it was a pretty straightforward jump for somebody to eventually be like, okay, well, what if we give you a very small controlled dose of this? Does that mean that you will be able to prevent reinfection? And this is before germ theory. This is before a real and solid understanding of how infectious disease works in the sense of how we understand it now. And yet, there's evidence that people were practicing this thing called variolation, where what they would basically do is they would take the scab off of someone who had smallpox, and they would then take that scab and they would make a little cut, an incision on an arm, and then they would put the scab onto that incision, bind it up, and then allow the body to deal with it. And so variolation was better than getting smallpox. No doubt, definitely better than getting a full infection by whatever means necessary, but it was still really brutal. Like, you can see here that this is from a paper that has a time course.
Is that a child's arm?
That is a child's arm, because they would do this mostly to children. Fun fact, the guy who ended up inventing the vaccine against smallpox, which was it was a cowpox vaccine, actually. But he tested it on his gardener's son, which if you look through the history books, or you look through Wikipedia, he's listed as his neighbor's son.
Children were the first lab rats.
Yeah, children were the first lab rats. I mean, the history of medical implementation is almost always the history of taking a vulnerable population and just being like, hey, we're gonna try this on you and see if it works or not. And that's often convicts, that's often military, that's often children, slaves,
psychiatric patients.
It's when you start to look too closely at it, it's very easy to get queasy. But I think that it's important to know that this is the arc of how we've always done this, and to know that it's something that is kind of an inherent aspect of developing medicine. And so in the future, this is something, and right now, this is something that we have to constantly be aware of how do we develop treatments in a way that's fair and just and ethical, that we're not just, trying it on whoever is most vulnerable. But basically, variolation, when you recovered from this horrific pox on your arm.
Is that gangrene in the center?
It doesn't look good. It really does not look good.
Gracious.
You would first of all have an enormous scar, right? Which means that basically a huge chunk of your arm was covered in scar tissue. Thank you very much. I just really want to get rid of the computer, A huge chunk of your arm would be covered in scar tissue, and then I think that his name was William Thompson. He was a biologist, he was a chemist, he was a natural philosopher. He noticed something. He was like, well, hold on. Those milkmaids appear to be immune from smallpox. And then he made the next leap, which is, well, if the milkmaids are immune to smallpox, and variolation keeps us safe from smallpox, then maybe the milkmaids are encountering something that protects them. And he discovered that they were getting something that was called cowpox, which is a related strain to smallpox. And so he took the scab off of one of these milkmaids, like an open oozing sore. He made a concoction of it, and then he injected it into a child. And the child recovered and didn't get smallpox. And so it was hailed as a victory. This was the first stage. I think he eventually tried it on himself, but he didn't try it on himself first, I have questions about that. But it basically opened the door to there now being a way of preventing this illness that has... I actually, what's the fatality rate of smallpox? Do you think you could look at this?
Yeah, I think this guy's name is Sutton, by the way, but okay.
So smallpox has a fatality rate of 30%. Much more contagious than COVID-19, much more deadly than COVID-19. You die in an absolutely horrifying way, as you can see from variolation. So anything that could decrease the incidence of smallpox was fantastic. The vaccines worked really, really well. Like, if you look at the incidence of polio in the United States over the course of the last 100 years, you see that after the vaccines were introduced in the 1950s, that you see a really sharp decrease in polio cases. Now, I want to say something here. There are perhaps other reasons that polio declined other than vaccines. And this comes back to the statistical sort of way that we have of looking at the world, which is that when we measure things and when we count things, it really depends on how we classify our measurements.
Case in point, there's a huge spike here right at World War One.
Right.
So why are there more polio cases at World War One, and why are there more deaths per polio case at World War One? Well, it probably has something to do with the war, which has nothing to do with the viruses themselves.
And the huge spike in the 1940s came after World War II, or in the late 1940s and 1950s came after World War II. And there is a very...
And after World War II came one of the greatest leaps in developmental progress that the Earth had ever seen. Wide-scale industrial agriculture,
clean water, clean air, clean food. It was an era of enormous progress, and so it's very, very hard to tell, is it the vaccine that has stopped polio, or is there something else that happens at the same time as the rollout of the vaccine that happens to decrease polio cases?
Probably both.
Probably both at once, right? Because it's obvious from this picture, if you go back here, that if you give someone a vaccine against smallpox, they get less of a disease than if they're exposed directly to smallpox. And so understanding I think that what's going to be really necessary in the future of medicine in order to make sure that our medicines are always effective and maintain their efficacy over time is to know when it is necessary to use a vaccine and when it is not. When it is necessary to use an antibiotic and when it is not. When it is necessary to use an antiviral and when it is not. And that requires us to have a really good understanding of cause and effect. And there may be more than one reason for why polio cases declined, aided as they were by the introduction of widespread global vaccination. We're at the point right now, down here, where the only polio in the world is actually vaccine-derived polio, where there's widespread vaccination campaigns, especially in Africa and India, have the side effect that they have allowed I think that there was a batch of vaccine that had some live virus in it, and so basically escaped into the population, and we have vanishingly small numbers of these cases, but they are the predominant representation of paralytic polio in the world. So vaccines are not without their problems, especially when you're using live vaccine, because if you have a bad batch, if some quality control fails somewhere, things can go off the rails completely. I think the battery died.
Yeah, it did.
Do you want to go get the charger? It's not a big deal. So on top of so vaccines are a huge boon to public health. They prevent the death from smallpox. They prevent the death from polio. We've deployed them for all kinds of diseases, everything from, the common cold to malaria to rabies. Like, rabies is a 100% fatal disease. If you get bitten by a rabid animal and you do not get treatment immediately in the form of a vaccine, you will die. 100% fatality rate. You will not survive rabies. And vaccines are the only way that we can survive rabies. So there's definitely a very, very hugely useful application of them. But vaccine hesitancy is basically as old as vaccines themselves. So in the 1970s, there was a preacher named Edward Massey who published a sermon entitled "The Dangerous and Sinful Practice of Inoculation." He says that the diseases are sent by providence for the punishment of sin, and that the proposed attempt to prevent them is a diabolical operation. then there was also the sermon of this other guy, Reverend Mr. Delafay, entitled Inoculation, an Indefensible Practice. It is very, very common for there to be this strain of what's called the naturalist fallacy, where people believe that disease is the natural state of man and that you get diseased because you are bad, and so interfering with the course of disease is interfering with God's will. And people can couch that in all kinds of different terms. There's all kinds of different mindsets about this, but it has been with us since the very first whisperings of vaccination campaigns. This was even the case when people were still variolating. They were like, well, you don't want to do that because you're interrupting God's will, which is crazy, because that produces a condition where you equate illness with some inherent germ of evil rather than with the fact that you have genetic mutations and you have illness and you have weakness of your immune system for external factors that has nothing to do with you. And yet we still live in a world where it's a relatively common effect. There's also this, I really enjoyed this caricature. So the first vaccines were taken from cows, and so this is called the cowpox, or the wonderful effects of the new inoculation. And so if you look at it, all of these, so on the left is a pot of, what is it? Hold on one second, I want to see the word. It says opening mixture. And so you have this line of people that are crowded into the door. You have somebody who's giving them ladlefuls of this opening mixture. This little boy is holding a canister that says vaccine pock from cow, and this guy is basically opening the wound and putting the cow pock into the wound, and everybody here on the right is immediately developing cow growths, right? So they have little cows growing from the pocks on their faces and from their ears.
This guy's got one coming out of his...
He's got one coming out of his butt. This lady's got one coming out of her mouth and from under her skirt. She looks like she might have like another one gestating. It's like a very, very visceral and horrifying image. And this is 1700s. So people basically right off the bat, you see these controversies that we have now. And then you start to look back in time and you're like, man, humans haven't changed. We still have the same fights. We have the same arguments. I can totally imagine somebody making a cartoon like this today with some like slightly different moving pieces. And so...
Yeah, I mean, it's not to say that it's like totally irrational either to behave that way, which is quite interesting.
Which is exactly what we get to next, which is the fact that hesitancy is deeply based upon the fact that vaccines did have issues. Live vaccines came from animal tissue. In this etching, what you see is the surgery table. There's a cow that's held down onto it, and this person is... No, it's a cow. It's a really poorly drawn cow. It's a very small cow, yeah. It's got hooves. Anyways, it's a cow. I think it's a calf. A calf. There was other pictures. It's obviously a cow. They didn't use, so we do use dog cells now, which we'll get to in a minute. But back in this day, we hadn't moved to dog cells.
No gloves during surgery back then.
Yeah, no gloves. No, not a lot of sterile technique. Babies in with everybody else. But basically, you would just like scrape the pustule off the cow. You would make a vaccine preparation from it, and then you would inoculate it into the patient, and the patient would go off on their way. And we didn't have a good sense of germ theory. This is before we even had that you should wash your hands. Like if you, there's this guy whose name was Ignaz Semmelweis, who was the first proponent of hand washing. He worked in a hospital. He was a doctor. And he found that there were very, very many women who were dying in childbirth. And so he basically went to his hospital staff and he was like, look, I think that if we stopped going to the maternity ward right after doing autopsies and not washing our hands, I think that we would stop killing women. And he was so ostracized for this that the man died in a
They accused him of disinformation.
They did. They accused him of disinformation, and he died beaten to death by a guard in a mental institution.
They were like, "You're so crazy that you need to be committed, sir."
And then,
But he
like,
was right.
and then, like 10 years later, they started washing their hands, and maternity rates fell. Right. So the arc of medicine also moves in these stops and starts where we do stupid things, and then somebody points it out, and everybody screams at them for how stupid they are, and then quietly later we come around and we're like, well, maybe.
I think, if I might, that the real solution here is to not be jumping to conclusions about either side as quickly as so quickly. A lot of these things take years to get to the bottom of. Medicine's side effects can show up years and years later. Still finding out about some of them, and so it's important to weigh everything with, and really to evaluate things non-violently too. Right? Wouldn't you agree? That's a real mess. Is the way that people talk about issues, even inside of science, sometimes is super violent. You know, everybody's calling everybody an idiot. This poor guy got thrown in a mental hospital. It's obviously not that bad anymore, but you'll get thrown off Twitter or something. So I think that we really need to change our attitude as a culture to at least, be able to tolerate arguments. I think it's really unhealthy not to be able to do that.
Mm-hmm.
No matter how stupid the argument it is, that should be even easier to deal with.
Mm-hmm. So the problem with vaccine hesitancy and people walking around and saying that vaccines are terrible and it's God's will that the sick should die is that it creates a polarizing effect in the discussion of the science, where whether you like it or not, you have to come, you cannot get away from the fact that there are issues with live vaccines.
Well, can I add one more thing? I'm sorry.
Course,
I just don't want to get away from this.
Please.
If you go back to this picture,
Sorry.
if we go back to the cartoon that we had before, these people are not wearing gloves and they're slicing with a dirty knife onto other people's arms. So it's very likely that there was all kinds of side effects of this procedure. And so these people who were scared of vaccines were probably scared for a good reason that had nothing to do with the underlying mechanism of the immunization itself. I just think it's really interesting.
Whether or not they realized it.
Whether or not they realized it, exactly. So, like, it might have, their hesitancy may have been very well justified, despite the fact that the medicine had great potential.
Absolutely. And that's exactly what we're going to get to next, which is the fact that live vaccines, which is where you basically have a virion that you have somehow deactivated, but it's still more or less the virion that you grew in some cell, they carry with them the risk of transmission of other things that are unintended. So early vaccines were maintained by something called arm-to-arm transfer, which is the fact that in order to have, you can't grow vaccines and you can't grow a virus in cell culture. You can't grow a vaccine
Viruses need cells to grow.
Thank you. You can't, like bacteria, you have a container of broth, you put your bacteria in the container of broth, and then 2 days later you have 10 billion bacterial cells. Great. You can use that to make a heat-killed bacterial vaccine against something like anthrax. But if you have a virus, you need to have a tissue culture system, and the easiest tissue culture system was arm-to-arm transfer. And so what they would be doing is that they would basically maintain sores on people's arms that were then used to generate
Human test tubes.
Human test tubes, basically, right? In order to generate the stock of tissue that contained the virus. And we're talking about the early days when there's no ability to purify the live virus that you want to use for inoculation away from whatever other contaminating pieces there are in that sample. So you have no bacteriological sterility, and your contaminated vaccines carry all kinds of diseases. So you can get tuberculosis, tetanus, syphilis. I don't actually know what erysipelas is. Do you know this?
I don't. But what's interesting too is that the contaminated vaccines could also carry things that worked in your favor. I remember from a couple of weeks ago we were studying influenza. they tried to make a vaccine against what they thought was a bacterium at the time, and they ended up inadvertently inoculating people against some sort of lung pneumonia bacteria
Oh, I remember that. Yeah.
that resulted in increased immunity, which was not stopping the flu, but since the people weren't battling this bacterial infection at the same time, it resulted in greater overall mortality outcomes,
That's fantastic.
which is quite fascinating. So it's not all bad news.
It's not all
But
bad
yeah, they were
news.
dirty. They were dirty vaccines originally.
And
And honestly, like even some of the vaccines we're going to look at today, the adenoviral vaccines are just that. They're adenoviruses, and
yeah, so we'll talk about
Yeah.
those in just a second. So
there's some concern that attacking these could be bad as well.
this picture over here is a piece of rabbit brain that was used to cultivate virus, and it's just in a petri dish on a square of cheesecloth. And what they're going to do is they're going to basically mash the brain up and then use that as the basis for their live virus preparation. But you can imagine that we're both mammals. The same way that you probably wouldn't go out into the world and grind up a bunch of rabbit tissue and then inject it into yourself, unless you had a very, very good reason to do it. This was the problem with early vaccines, which is that they weren't, while they cured the disease, they also carried side effects. And for many, many years, it was difficult to talk about the side effects because the sort of, it's God's will that you should die, and so we shouldn't have vaccines crowd was an effective mask for legitimate problems with production techniques. This picture is not particularly relevant to the discussion, but I love it so much.
I was thinking this would be a good opportunity for you to tell your favorite biblical story about the God needs you to
Buy a lottery ticket?
Yeah.
What's the reference?
Oh, that you have to help God out a little bit sometimes. Tell the story. Come on, it's
Yeah.
awesome.
I don't. So there's a Russian anecdote, which is the fact that, or I guess it's Russian Jewish.
This is a Catholic school, so
Yeah.
we're allowed to tell these stories.
So Yitzhak he's this very observant Jew, and he's always he goes to synagogue every day. He observes the Shabbos. He keeps kosher. He is the best servant of God that has ever been. And, you know, he looks around at his life and he realizes that he's like, man, life is hard. And I wish that I wish that there was some reward for this work. And he's like, man, I know that there shouldn't be reward for this work because I do it just for God. But he prays every night and he's like, God, you know, I believe in you. I have faith in you, but I'd really like to win the lottery. And if you exist, please let me win the lottery. And so he prays like this for a long time. Nothing happens and he's getting kind of sad. And then one night he's sleeping and he dreams that he's in front of the burning bush. And he again, you know, falls to his knees and he prays and he's like, please God, like I am your greatest servant. Please let me win the lottery. And then the voice comes from the burning bush and it tells him, Yitzhak, go buy a lottery ticket. Which is to say that, like, it's not enough to just talk about stuff. You have to move in the right direction. And you have to take the steps that will protect you. And there's this, I don't know if this is actually from the Bible, but this is just something that my mom would always tell me, which is the fact that God protects those who protect themselves. And vaccination is the method of protecting yourselves. And you have to be able to look critically at all of the structures that you are using, and you cannot simply close your eyes and just keep your hands on the wheel and basically hope that you'll be able to drive all the way home. You have to have your eyes open, you have to be looking at what's in front of you, and you have to be careful. And with something as intense and as life-changing as vaccination, that's definitely you should be looking at it. And society often has these ways of not being able to look at it because it's an unpopular opinion, or it's an opinion that carries unpleasant connotations, or you don't want to impugn the validity of the science. And so you end up with cleavages where people can't really talk through what should be talked through. Was that your lead into that?
Yeah.
I just love this picture of Elvis. Elvis is getting a vaccine. Elvis was like, this is a very common thing where it's like, because there is vaccine hesitancy.
Do the kids know who Elvis is
Yeah,
these
do you guys know
days?
who Elvis is?
Elvis is like my dad's favorite rock star.
Well, we got nods. Okay, that's good.
He's
I
the
wonder if
king.
I wonder if there's ever going to be a day where they don't.
Maybe. I don't know.
I don't know. I hope not.
know.
But so this is a very common thing. Like, I don't know if you guys saw, but on the Colbert, there's a huge number of TV shows and network presentations during COVID, which were intended to get people to take the vaccine. Did anybody see the Colbert report or like Colbert hosts some like nightly news thing, right?
I'm out of the loop.
I don't know. Colbert does something, and there was this like big thing where he had like a line of dancers and he sang a song, and it was called like the vaccine. If you can pull this up, we'll play it after the break or something. But there's always been this sort of push and pull where
Just
people
the Late Show now.
Yeah, the Late Show, that's right. People are hesitant, and so there's this push to use figures that they trust in order to create acceptance, for better or for worse. So we've moved away from live vaccines that are isolated just from the skin of animals, and so now we're using cell culture. To kind of wrap up the last bit before we move to the papers, which is that when we moved away from using live animals to produce our vaccines, we moved to cell lines. But cell lines also come from animals, and so we use these maiden Darby canine kidney cells. We use Vero cells that are the kidney epithelium of African green monkeys, and then we use these human cell lines. Human cell lines are really weird because, I mean, all cell lines are really weird, right? Because you isolate cells from a specific creature and you more or less continue to use them across the board in many, many, many laboratories. So these canine kidney cells come from a single point of isolation. They just isolated a buttload of them, enough to be able to distribute them globally and use them in massive quantities of immunology research. But Am I right to say that there is a finite supply of these cells in the world?
Are they immortal?
I don't think they're not immortal.
How do they live then?
I think that they have frozen stocks that they thought they have like a set number of passages before they become exhausted.
I'll figure it out if you want to go on.
So these are all also not ideal systems because you're still in a mammalian system. If there are any kind of contaminants in the Vero cells or the maiden Darby cells or the human cells are mostly isolated from fetal tissue, which is also another huge ethical morass that you get into, where basically you're using this fetal tissue that's isolated from either stillborn or aborted fetuses, which are then just distributed into the population eternally. It's another one of these things that people kind of look at.
So they proliferate when they're exposed to collagen. I guess your kidney tubules are continually regenerating in response to collagen.
So they just add collagen and then they get new ones?
They make little sandwiches out of them, yeah.
Fascinating. Okay, so they don't run out. But basically, this is a case of using a system that is better than live animal, right? Because we know that live animals can carry diseases that are able to transmit it to humans. If we move into cell lines, we're able to at least constrain the variability because we're using a very set collection of cell lines, but you still have problems. You still have endogenous retroviruses. You still have transposons. You still have these things called exosomes. You still have mutations. You have all of these natural products that the cells are making, which you cannot always purify away when you are purifying your desired viral particle for immunization. And so that kind of brings us to today in the sort of the 30,000-foot view, which is the fact that moving to DNA and mRNA vaccines is the ideal solution to a problem that has been plaguing vaccination since basically the 1700s, which is that if you can use purified nucleic acid information to generate immunity, you have greatly limited the potential dangers that can be carried along in the vaccine.
However, you might be introducing other interesting complications too, because by feeding a cell raw nucleic acid, you have the potential for non-specific interactions and genomic incorporation and all sorts of fun stuff like that.
Right. And so we'll kind of get into that. But I think that at this point, there's what? Sorry. In addition to the cell-based vaccines, there's also egg-based vaccines where they take basically developing egg embryos. So they're fertilized and they have the baby chick inside of them. They inject the antigen into the eggs, or they inject the virus into the eggs. The virus proliferates inside the eggs, and then they're able to isolate the inactive virus from inside of the eggs. This, again, you're really looking at these systems that are doing kind of morally questionable things on a massive scale. And you can carry over various unwanted side effects from these egg-based vaccines as well.
I mean, a lot of these things are real moral conundrums because there's no right answer. It's a matter of minimizing harm, and oftentimes, side effects are a really ubiquitous problem in all of medicine, I would say. I mean, even if you go in for some routine surgery, it's possible that something totally awful could happen that's unexpected, even a root canal or something.
Mm-hmm.
So it's not so much that you're ever going to be able to have zero side effects. It's more a question of, are your side effects less than the disease itself for a given population? And for a given population is really what the second paper is really concerning itself with. So,
And so from here on out, I think that we just have different vaccine strategies, which might be
let's
best.
do that paper.
Should we take a 5?
Sure.
Yeah, let's take a 5. Let's come back at 44. All right, let's do 45. Let's do 45. See you in a few minutes.
All right, so we're going to start with the overview of vaccine development paper. And we've talked about some of these things. We can jump over to the figures too. And then we're going to briefly touch on the second paper, which talks about the strategies for application of these vaccines. So obviously there's 3 major pharmaceutical companies right now, primarily Pfizer and Moderna. Moderna is combined with another vaccine company that's slipping my mind, who have taken the prize for this. But let's go through all of the strategies. Paper starts off mentioning that normally in the vaccine development process, we're talking about years upon years to pull this off. And so they briefly go through the trial process, and how this has really been a ramped up effort lately. I think my headphones.
Right. So I was actually just looking at this today. The normal arc of vaccine development is 5 to 10 years. And that involves all phases 1 through 4, with basically phase 1 actually is usually the longest period of time because you give the vaccine to really, really tiny numbers of people, and then you really closely study long-term side effects. You basically look at them for 5 years after giving them this to make sure that nothing happens.
So on one hand, it's been kind of amazing that we have pulled off a vaccine so quickly. I think a lot of that had to do with advances in genome construction sequencing. We had a target for it essentially within a month. If you want to go back also, can we go over to the slides real quick? I pulled the slides I think a lot of it also has to do with this world we live in that's driven by technology. This modern vaccine was developed largely, I didn't have a good comparison, but it was developed almost entirely in the private sector, which means that there was huge money available from investors to be able to pull off this work that usually takes 5 to 10, maybe even 20 years. I don't know where the slide is. Do you? There we go. So if you look here about where the development costs came from, it's almost entirely from the private industry. And so this is, on one hand, pretty awesome because it's allowed a huge amount of work to get done really quickly. Everybody who's ever worked in government will tell you that things that happen in the public sector happen very slowly. things that happen in the private sector happen very fast because you get investors who are willing to take a risk and give you a bunch of money, which means you can hire more people and you get more work done. So on one hand, this is pretty awesome. On the other hand, it's a little, you're also sacrificing some of your safety, or what should I say? It can be risky too because you're entrusting people who have a vested interest in seeing it work out to deliver a product to you that you're depending on to not have these longer term effects. So it's a really wild, multivariate risk benefit calculation that we've been watching unfold. And I think that's why there's so much political turmoil about it too.
It doesn't help that 45% of the FDA's operating budget is paid for by pharmaceutical companies.
Yeah, and all the trials, like we talked about last week, all the trials are being paid for, not all of them, but more than 75% of the trials are being constructed and paid for by the companies as well. This is like not to say that it's an entirely a messed up thing. It does accelerate development and innovation, no doubt. So it's not a simple black and white issue like pharmaceutical bad, public research good. It's not that simple. It's an extraordinary accelerator, but we need to develop ways of checking it better too. So
it's an extraordinary accelerator that is not particularly transparent.
Exactly, yeah. So, with the innovation of new transparencies, new registries, new ways of homogenizing data entry and analysis, maybe even public analysis, blockchain level transparency, there's ways to make this safe while retaining all the wonderful things we love about the free market.
Not to put you on the spot, but what would the blockchain do?
The blockchain?
Yeah, what would the blockchain do?
Transparency for data insert and analysis. Yeah.
All
right. So that being the case,
should we go back to the paper?
Yeah, back to the paper.
All right.
So the first one they talk, or if you have a thing open for live vaccines. Do you have
well, we kind of talked about live vaccines.
Okay. So, interestingly enough, there are some live weakened vaccines being developed. However, the live vaccines are actually against leprosy and tuberculosis. This is similar to the influenza vaccine that attacked the
1918 pen?
What's it called? Pneumonia bacteria and increased immunity. So this vaccine is called BCG, which stands for Bacille Calmette Guerin, which has been used to prevent tuberculosis and leprosy since 1921. But I guess in preliminary studies, they found that it actually decreased SARS-CoV-2 infection rates.
But not directly.
Right, not directly.
They think that the mechanism of action here is that I actually do have a slide for this, that what the BCG vaccine is doing is so basically what they think that it's reprogramming the monocytes, which then start to produce the necessary inflammatory markers and are able to boost the immune system. And the boosting of the immune system, they think, is making it more efficacious against SARS-CoV-2. But as you can tell, there's a question mark, and people aren't exactly totally sure if there is some kind of shared pattern that's on the BCG vaccine versus the SARS-CoV-2 vaccine, or if it's some kind of other mechanism of action. A lot of this stuff is very preliminary, as it is with a lot of the repurposed drugs, because people just try stuff. Some of it works, some of it doesn't.
Yeah, so they've got a few trials going on. As of the date of this paper, they had a phase 3 trial going on with it. Actually, looks like they had more than one. They had a healthcare staff trial too going on in the Netherlands. They have another one, which is an avian infectious bronchitis virus. A chicken coronavirus. And similarly to what Dr. Beneberry just mentioned, it seems to have indirect effects and maybe useful in COVID-19 as well.
Well, it's interesting because they think that it's beneficial for SARS, not because it's activating the immune response. They think that it's blocking the production of antibodies and other immune responses, which would suggest that It's the opposite mechanism of action from BCG, where BCG would be increasing your ability to clear the virus versus this one would be increasing your ability to not freak out when you see the virus.
Interesting.
So
Yeah.
very different approaches, but to the same end, using the same technique.
Yeah. I wonder if the introduction of antibody or blocking antibodies is a good idea. Certainly, blocking out of control immune responses makes sense. That'd be an interesting paper to follow up on.
Seriously.
Anyways, they just make a one line. It might be interesting to be investigated as another choice. Uh, doesn't look like anybody's actually doing that right now, according to this paper. Then we have DNA vaccines. DNA vaccines are just what they sound like. They are going to actually tell the body to make something like spike protein. Do we have a slide for the DNA vaccines?
I did not, but there's that really good Wikipedia one. Should we look at that?
Yeah, sure. So I have a chart also that shows we'll do that.
There's a
chart.
couple of different problems with DNA vaccines. So DNA vaccines, you can make it against any you can make it against any protein, but you run the risk of it messing around in your cells in a way that you might not like. So, you want to walk through it?
No, go ahead.
So basically, you have a virion, you have a virus, a viral program, and it contains genetic information, and that genetic information is going to encode proteins. In this case, we're looking at West Nile. So you have a cross-section of the West Nile virus that contains within it RNA. But rather than using the RNA that is the same as the West Nile virus as the desired substance for the vaccination, what you do is you isolate this nucleic acid from the destroyed virus. You decide what genes it is that you want to use, and then you reverse transcribe the RNA inside of a test tube into DNA. Once you have the DNA sequence, you can synthesize it, you can isolate it, you can amplify it using PCR, you can get massive quantities of it. So you can start with 10 virions and end up with a stadium full of PCR product. So this is, I haven't seen a DNA synthesizer that looks like this, but basically once you synthesize all of the PCR product that you need, you have this fragment and it's called cDNA. It's called cDNA rather than regular DNA because it's complementary DNA. It's to signify the fact that you've isolated it from RNA rather than isolating the DNA itself. But you take this DNA, you put it into a plasmid, you grow lots and lots of plasmids in bacteria. So basically what you do, I don't know if anybody's done protein purification, but this is basically how the production of things like insulin works. If you're ever doing any kind of protein crystallography or you're doing any kind of protein characterization, this is what you're doing. You take a vector that contains a gene that you want to have transcribed into a protein, you inject it into bacteria, you grow the bacteria until they're really dense, then you turn them all on so they start to make the protein, then you spin down all the bacteria, get rid of all the gunk, you take your protein, you purify it, and then, in this case, they're not even going all the way to protein, right? They're just going to plasmids. So they're just cutting the plasmids out and then using those plasmids that carry the genetic information. That's what's actually going to be the vaccine. You just inject those.
And so DNA obviously has some advantages over RNA in that it's more stable, first of all, which means that you can pile it up and transport it a lot easier.
mRNA has to be refrigerated, which is why distribution of these vaccines has been so slow around the globe.
It's a lot easier to make than some of these killed type viruses. Both RNA and DNA. We're gonna get to RNA next. Maybe we should have done it first, but they're both. I think you mentioned this earlier. They're both much cheaper, and they don't bring as many variables into it. You don't have to have animal cells. You don't have to have animals to grow it in. You don't need weird chimeric hybridoma type things.
It's almost abiotic.
Yeah, that's true. So ideally, it's really nice because it's just one ingredient, and that's when it comes to medicines, when you can really isolate things down to individual, even when it comes to food, like if you look at the back of your wrappers of things, it's like probably the health is like inversely related to how many, inversely related to how many ingredients are on the back of the food, right? There's all these chemicals and stuff. It's like, what am I eating? And the same thing goes for drugs too, and vaccines. You really want to keep it as simple as possible. So both of these RNA and DNA pull that off really well. DNA, however, poses some particular disadvantages because that's obviously what your genome is constructed out of. And so and not only your genome, but the genome of bacteria and other parasites that might be present in your body. And you have the possibility of essentially transfecting, getting that DNA moved into the genome of the host organism. This is particularly scary because a lot of vaccines for respiratory viruses, the main delivery system outside of using shots, which we're all familiar with, is nasal sprays. And so there's a lot of concern that DNA nanoparticles could go right through the nasal lining and infect the brain very easily, transfect the brain, I should say, and actually end up in the genome of non-target cells.
And
so
this is actually the mechanism that's used for immune treatments. Normally, and this is a specific case we have here. This is for a DNA-based treatment for CAR T cells, but you actually can have plasmids whose function is to integrate into the DNA, and it's possible to have off-target effects simply due to systems that we don't know yet that at some low frequency are able to do this even though they're not designed.
It's a CAR T cell.
It's something cancerous.
This is like they're just trying to modify some T cell here.
Yeah, they're trying to modify a T cell.
This is not part of the vaccine.
No. This is just an example of recombination of the way that you would take a plasmid, and using transfection, which is what you basically do with a DNA vaccine, is that you would end with it integrating into the DNA because the plasmid carries these necessary inverted terminal repeats, and it carries the enzyme that does it, which is the transposase. But you can imagine a situation where somebody has transposase available and the sequence inside your viral vector has these inverted terminal repeats, and you can end up with off-target integration. That's kind of the point that I'm trying to make, which is why DNA vaccines up until this point have been developed but never approved, except for they just approved one for emergency use authorization in India for COVID.
No kidding.
So we'll see how it develops.
And I assume that's like if they run out of everything else, something like that.
I think that it's just one of the main vaccines that they're using in India.
That's wild.
Right?
Yeah, let's keep an eye on India, see how that goes. So the RNA ideally bypasses this danger because, well, this is a great chart, because RNA is not going to go into your genome by itself. It could get reverse transcribed into your genome, maybe, although that doesn't seem to be much of a problem according to the people that are studying this. There are a few papers suggesting that it is possible, so we'll keep an eye on that as well. I think I linked one or pulled it up. For next week. Yeah. For next week. So we're going to talk about long COVID, and I think that one of the papers we're looking at is not about long COVID, but it is about a hypothesis for why you would remain PCR positive long after the infectivity stage is over, and if that involved reverse transcription and embedding of the viral genome into your DNA. So we'll deal with that next week. This is a great summary of really the treatments that we've talked about.
There's one thing about DNA viruses that also makes, or DNA vaccines, which are just plasmid-based vaccines, a little bit less effective, is that you have to use a special tool to get them into the body. You actually have to electroporate cells, which means that you have to use an electrical vaccine gun. That sounds terrible, but it really isn't. It's just when you give the vaccine, you also apply a very small electric pulse, which forces the cells at the vaccine site to open their membranes up a little bit to let the DNA in. Otherwise, there's no incentive for them to allow the DNA to come into the cells because they normally would not pick up just random DNA that's on the outside.
I guess that's why they use nasal sprays for them.
Might be.
They have to be delivered at the site where you're trying to create the protection. Right, so next up is RNA vaccines, which we've talked a fair amount about so far. And these are obviously the ones that we have settled on. They have been shuttled through the various uh, phases of clinical trials. They don't really say much about them here.
They do later. Later
Okay.
in the paper, they come back to them. It's like the last section of the
Okay, let's
paper.
come back to them ourselves then. Some of the other ones that were tried out, subunit vaccines. Do we have a slide for this?
Sure do.
Cool. So subunit vaccines, also called virus-like particles, are this interesting idea where you make a sort of collage of all of your favorite viral proteins and hope that that will induce the response. What's up with this figure?
Basically, what you do is you create a virus that doesn't have any nucleic acid information because that renders it as being harmless. If your method of viral danger is correct, which is the fact that it is the replication of the virus and the presence of this exogenous nucleic acid material inside of cells, then you immediately remove that problem by having these virus-like particles because they don't have any of the nucleic acid. So only the amount that you inject will be the amount that is ever present in the body because you're not allowing the body to produce more of them. They're basically a one-time boost. And the way that this works is that basically you produce this all of the proteins in various platforms. Over here you can see that it's possible to use bacteria, yeast, insect cells, mammalian cells, plants. You collect all of those proteins. So you basically lyse the cells, pull the proteins out, and then you mix the proteins together. And because of the way that viruses work, which is that their surfaces are like jigsaw puzzles, you spontaneously assemble a virion that doesn't have any DNA in it, presumably, hopefully, because, right, if you have any contaminating DNA, it is possible for chunks of it to get in there. And you can have lots of different, like Dr. DeLay was saying, you're not stuck with the antigen of a single protein. You can make a virus-like particle that looks exactly like SARS-CoV-2, but you can also take a tumor antigen and put it on the surface of a virus-like particle, and then use that as an immunotherapy against cancer.
Now, the tricky thing about these is that they are only going to be passively distributed. I worked in a lab at one point that was working on these sort of particles against tumors, and it's very difficult to deliver them to the site, especially to get them deep inside of a tumor. Unlike viruses, they're not going to replicate, so they're not going to just daisy chain themselves down into the tissue.
But I can imagine, for example, it's been discovered that you can target probiotics to the center of tumor cells. And so if you could have a bacteria... Sorry, did I say antibiotics or probiotics?
You said pro.
Pro. That's what I thought.
Because they walk down there or something?
That was what Tal Danino at Columbia was doing. He was basically coming up with like targeting proteins for cancers.
Yeah.
Targeting bacteria for cancers.
I see. Well, yeah, anything alive could be able to walk. This is bordering on xenobot type stuff.
Yeah, and so if you can have a bacteria that... He was designing these really cool systems where, let's say the bacteria is capable of producing this virus-like particle, and you allow it to express proteins inside of itself for a long time, and then you break it open, and the antigens are present right on the inside of the tumor in this tuned delivery format that uses things that are already in the body. And so they're pretty cool systems. I think that these are some of my favorite approaches just because it's abiotic, there's no nucleic acid available, it's produced in a very clean way, and I think that it's a very responsible way of producing it because you can use bacteria and yeast to do it rather than relying on mammalian cells.
I like that, yeah. Another thing that's really cool about it is they can... One of the groups, I forget which one of you, is looking at binding kinetics using MD simulations where you actually see what part of the lock and key system lines up. And they're saying here that in this paper that several groups are able to begin to look at how B cells bind, what part of the spike protein they bind to, and they can actually engineer, they're beginning to engineer these proteins, these vaccination, what do we call them? the virus-like particles. So the different epitopes that are on the surface of the virus-like particles, they can custom tailor those to excite cytotoxic uh, lymphocytes, T cells, B cells, uh, and so that's pretty cool. So they're real designer molecules. Or this is a real designer vaccine. It's pretty
Yeah.
cool.
And I think that designer drugs are kind of the future anyway. So it's
You can even imagine that if they were able to look at your own body, they could custom tailor these in the future to line up to whatever your particular T or B cells would be most excited by. So yeah, I think those are pretty cool too. Are there any trials going on? Oh, they've also got. I wanted to mention this too. Uh, they've just. I don't know if we've talked about this, but there's these things in these vaccines called adjuvants, which is a really uh, nebulous uh, term.
it's a helper
It's
molecule.
a helper molecule. It means almost anything, but it's something. It's some general broad-based molecule they toss into the vaccine because it helps elicit an immune response. I remember asking my immunology professor about this in grad school and having him just basically be like, "Well, it's just whatever," and I was like very dissatisfied. But they found essentially certain amino acid sequences that strengthen the immune response. In other words, tell your body to be alerted to the fact that there's something over here that we just injected into your arm. Come take a look at it. And they're getting better and better at those. So that's what a Padre sequence is. And yeah. Cool
Then we
stuff.
have these. Sorry, did you have more to say?
Yeah, let's go to vector-based vaccines.
Vector-based vaccines. So vector-based vaccines are kind of what we've been talking about in terms of these.
Vaccine is sort of like taking a virus that you're not too worried about and repurposing it.
That's exactly. Okay, thank you. Or just my brain went blank and how to put that into words. So there's this virus called an adenovirus. And adenoviruses are widely accepted to not be dangerous to humans, which means that they can go into your body.
40% of you are seropositive for it right now for adenoviruses.
And they do apparently nothing. There's other viruses that do this. There's like tupanoviruses. Do you want to look it up? That'd be cool to know. So because these viruses basically have a free pass to get in and out of cells, you can introduce viral DNA by using the adenovirus as a vector. So you basically take your nucleic acid information and instead of sticking it into a molecule of SARS-CoV-2, you stick it into a molecule of adenovirus. And instead of giving the full genome, you just give the gene that you're interested in looking at. In this case, you would give a spike protein. And so the adenovirus gets into the cells, it's able to produce the spike proteins get chewed up by the proteasome, they get displayed on the surface by the MHC, and then the T cells can notice them, and then you're good to go.
So there is some concern too that we don't really understand adenoviruses, and based on the fact that most of them do absolutely nothing pathological to us, that immunizing people against adenoviruses could offset the natural balance in the body in some way too. I came across this. I'm not sure if it's in this paper. But the idea being that if your body starts attacking all of your adenoviruses because of the vaccine, that could be a problem as well.
It's antigen creep, right? So it's like a Pavlovian response. If you associate an antigen that should be responded to with an antigen that is totally neutral, it's possible that you could train the immune system to start to react to the neutral antibody or the neutral antigen. And once you do that, then 40% of people suddenly develop autoimmune disorders because they already carry adenovirus. And we don't know what's going to happen if the immune system starts to get really reactive to them.
Well, we do know what happens if, in a worst case scenario, you get a cytokine storm type situation. But they don't really want to find out if that's going to happen across the population. So they're to be, all of these vaccines have pluses and minuses again. So we're talking about minimizing those minuses and expanding the pluses. And it seems like we err on the side of safety, which is kind of cool in our society right now. It's hard to say because we didn't none of these papers look at the efficacy across all of these different platforms, but it seems like they are generally favoring the nucleic acid vaccines because are they have less potential for complications, safety complications,
Sure.
at least in the short term.
And I mean, it's like, I, I think that it's very important to disabuse ourselves of the idea that it will one day be possible to reach a place of no consequences. That one day we will have a perfect method that has absolutely no side effects and has no drawbacks and creates no other problems in the universe. This is not how things work. We live in complex systems. A butterfly flaps its wing over the Atlantic and there's a hurricane. There's a typhoon in the Pacific. There's a massive flux of the way that these things are all interrelated, and when you start changing one thing, you're going to run into problems down the line that you couldn't have even possibly imagined. And when you fix those, the same thing will happen. That's how evolution happens. It's the fact that you're constantly in conversation with your environment rather than settling at this stable fixed point where nothing changes, because that,
And
I
that's why
think,
you
is
want to
death.
minimize the variables too, I think, which is really cool because, like, let's say worst case scenario that these mRNA viruses have some sort of longer term consequences. They're somehow getting reverse transcribed or something. Okay, well, even if that it does turn out to be true, like 50 years from now, then that's one problem that we can deal with. We've still minimized the set of all problems that we have to deal with. We're not dealing with other viruses creeping in from mammalian cell culture. We're not dealing with other off-target effects. We're still just like, okay, now all we need to do is stop these things from being incorporated or whatever it happens to be. So we're always kind of constraining our set of problems that we need to mess with. And I think that's a huge part of why people went with the RNA viruses over some of these other possibilities is it does constrain the set of potential problems. It's not saying that they're necessarily without problems, but it does constrain the set of problems.
Mm-hmm. The last thing here that they say is really insane. So it sounds like they made an artificial antigen presenting cell using genetically altered lentivirus. So that sounds like what they were doing is that this is a cell-based vaccine that was developed by infecting cells with this lentivirus that carries SARS-CoV-2 genes, and then the cell begins to present those antigens on its surface, and then I think that they inject the antigen cells. Right? So they're altering dendritic cells with these lentivirus vectors and then injecting the dendritic cells.
So what dendritic cells are the cells? They're like the surveillance cells in your body. They will go out and grab all the pieces of junk and bring it to the lymph nodes so that we can make antibody with the whole B cell, T cell interface.
That's fascinating. Then we move to these other vector processes. So something that you guys have probably heard about is this ChAdOx1, which is the Oxford Biosciences vaccine. We haven't seen it that much here in America. I think that it's more popular in the UK. But again, it's using the same adenovirus vector that carries the SARS protein. So you basically have a virus packaged with this specific protein, and the functionality is the same as the mRNA vaccine. They both produce spike protein. The only difference is the method of application. And then there's other adenovirus. We won't necessarily go into those.
I think we should jump over to the second paper.
Yes. Let's just look at this really quickly. So this covers all of the different kinds of vaccines. Live weekend, which we've talked about, DNA vaccines, which are DNA molecules, RNA vaccines, which are RNA molecules, subunit vaccines that are assembled out of many pieces that don't contain any genetic information, vector-based vaccines that are basically just carrying, that are deactivated viruses or harmless viruses that are carrying a single gene. And then there's this inactivated SARS-CoV-2, which is basically just digested or heated virus. The way that they deactivate it is chemical or physical methods, right? So they don't actually say.
In one case they stop it from creating one of its envelope proteins, so
Mm.
it can't reassemble.
So it
They
basically
take the
goes through
gene out,
one
I guess.
process and that's it. I mean, I think that the takeaway from all of this is the fact that there's a lot of different techniques that people are using to be able to start making these vaccines, and there's many different directions that people can come at.
And yeah,
And then let's go through this one quickly.
hopefully we've more or less answered the question of why the world is settled on these RNA vaccines as well. Uh, they're, you know, concerning the set of possible problems, not necessarily without their own problems, but we're getting closer. Of course, vaccination itself has some serious problems. That's what the next paper is mostly about, is actually about distribution, because obviously the vaccines are chasing a moving target. And that being the case, how do we best, what's the strategy for applying the vaccine? Who should it be going to? You know, this paper starts off right out of the gate, and it mentions, this was kind of shocking to me. I didn't realize it, but the greatest coverage that they can even hope to achieve in 2021. This paper is from 2021, isn't it? Is 17%.
Coverage of vaccinated individuals around the world.
Yeah. Okay, so they're like, look, uh, there's tons of vaccines, but there's still only enough to reach 17% of the population with the scale of production that we have right now. Remember, RNA is very unstable stuff. Like, if we were to just throw some RNA on the table right now, it wouldn't last for more than a few minutes. You need to be constantly producing it in order to distribute it and reach lots of people.
This thing about the 17% is specific to Africa.
Yeah, that's okay. Fair enough.
And Africa has the problem of vaccine distribution, where because you have to have refrigerated vaccine, there's a lot of severe limitations to being able to get it into rural areas away from the big cities.
Right. Africa, what's the population of Africa? The percentage of it, though? It's not a small part of the world.
No, it's not a small part of the world, but the point is that the argument that this paper is making is that there are conditions where applying the vaccine is super important and should be done, and there are conditions where perhaps it is not as necessary, and that in order to help our stocks go farther, in order to help our public health programs be more effective, we should be treating in a smarter way than we have been to this point. And that's basically what this entire
Work harder, work smarter,
about.
not harder.
Work smarter, not harder, because if you're spending less vaccines in your super wealthy countries, you can use those extra vaccines and you can go to Africa and you can go to India and you can go to Asia and you can really immunize people and help the world a lot if a single dose is enough. And basically this entire paper is making the argument that, hey, maybe a single dose of this vaccine is going to be sufficient.
In most of the population, particularly in places like Africa, where, 85% of the people have already been exposed to the infection. So they basically I don't think we actually have time to go through this whole paper today, but they basically make the case that if you've been exposed to this virus, you're going to do much better after receiving a single dose than somebody who's even received multiple doses without having been exposed to it in the first place. And that's a really important piece of information that needs to make its way into public health so that the supply can reach more people. Because if we're doing this to help people, then it doesn't really matter where those people are. We need to make sure that we're meeting our goal, which is to help people.
Mm-hmm.
And, so it's not clear that we should be wasting 4 doses of a vaccine on somebody who is not going to do as well as somebody who's already been infected in one of these other countries, or even in your own country.
Or the vaccines aren't actually stopping transmission. So I have a couple of highlights that I'd really like to get to, and we have about 10 minutes left before we have to switch over to questions. So let's go through some of these key findings. Number 1, they make the case that multiple vaccinations don't stop asymptomatic transmission. And this was a major argument for the rollout of the vaccines in the first place, was the fact that it was going to stop the pandemic in its tracks. But what we found is that perhaps that was true for the alpha variants. The variants that have emerged in the aftermath of the vaccination campaigns don't have this case. In England, 40% of the infections in fully vaccinated household contacts came from a fully vaccinated index case, which means that somebody who is totally vaccinated is getting a case of COVID from somebody else who's totally vaccinated. This is not uniform across countries, and so that requires some analysis, but it does suggest that full vaccination or multiple vaccines aren't going to stop transmission.
I think it was even worse in the Netherlands.
It was better in the Netherlands, 63%. Vaccine effectiveness was 63. They reversed their statistics. It's so strange. So, they are making the point that
it's another thing to mention too is that those stats are going to change too as time goes on, right? Because if you do all these studies when you have one variant and your vaccine is modeled off of epitopes that concern one variant, but then that changes, then you're going to get a totally. These numbers are probably going to get worse as time goes on. Is I guess what I'm trying to say. Because as that virus mutates, it's going to be even less effective than it is in this case.
So now we get into the fact that they're like, look, you're not going to be able to stop this. It's just not going to happen. It's going to become endemic, right? So they're like, in the post-vaccine world, aggressive containment strategies, including border controls, will have reduced benefits. They likely discourage global data sharing. And so these fear-driven mechanisms of just close the doors, don't let anybody through, we're going to figure this out on our own, don't work. So they're like, let's prioritize giving single doses. Why do we think that we should give single doses? Well,
hold on. Why would closing borders this is really interesting. Why would closing borders discourage global data sharing?
Do you have a vision, or are you?
I
Is this a rhetorical
don't know.
question?
I do think it's interesting. I wanted to brainstorm a couple of reasons. It makes I can think of some.
Well, you'd punish a country for sharing the fact that they had a new variant if you're going to basically block travel to that country in any kind of.
Right. Yeah.
That's what I think.
Yeah, that's it. And I think it's interesting too because you see this in the doctor's office too with insurance stuff also all the time too, where they're like, you have this patient-doctor confidentiality, but if it affects your insurance, it's not really that confidential. So you get people reporting lies basically, which then hurts the overall meta-analysis later when we're trying to figure out what causes people, like what their dietary issues are that cause disease. Well, if people are lying about their diets because they're afraid of their insurance like being ripped out from under them, then you run into a real mess. And I think it's the same way with the self-reporting from viral statistics. So closing borders is not a healthy thing to do.
So they now go on to justify why other strategies are better. They point out this really interesting thing where they basically they're like, look, so even though there's only 21 days between these vaccinations, there's pretty good single dose efficacy for COVID. So you have basically 60 to 90 single dose efficacy from the Pfizer vaccine.
In that window.
In that 21-day window, 75 to 80 for, I think this is the Moderna vaccine. And this is also the case for these adenoviral vector vaccines that we talked about.
And these, by the way, were the efficacies that were reported initially that led to all of the mandates and stuff, where people were like, well, they're perfect. They're going to lead to 90% efficacy and all of this stuff.
Sure. And what they're basically showing is that you can actually expand that, because in a Canadian trial, they saw that the highest efficacy against the virus was if you expanded the gap between boosters, between vaccinations, to be 35 days rather than 21 days. And then they showed that there was actually a case where with the COVID vaccine, with the Pfizer vaccine, you can expand that to 60 days and then you get even stronger immune responses. So they're building their case. They're like, look, if you expand this period between vaccine administrations, that gives you the ability to give one vaccine to everybody. And then you have 2 months to boost production by the time they need to get people their second vaccine. So maybe that's a smarter way of doing things. They're saying that you have protection for nearly 10 months, that whether or not antibodies were present post-infection. So that's interesting. But what I really want to get to here is they're like infection naive recipients of 2 doses of the Pfizer vaccine were more than 7 times as likely to develop a Delta infection than individuals with a previous infection, among whom a single Pfizer dose gave an additional 50% reduction in infection risk. I went and I dug up this paper. And this paper is very cool because what it shows is somebody asked at the beginning about hybrid immunity and how hybrid immunity works. So what you have here is you have 3 different groups. You have those that have just natural immunity. You have those that have been vaccinated and gotten a booster. And then you have people that have hybrid immunity, which means that they have recovered and then they have gotten the vaccine, or they've gotten the vaccine and then they've had COVID. And what you can see here, these error bars are actually quite nice. And the effect size is huge too. So you can see that when you get vaccinated and a booster shot, you have a brief period of protection right after the booster for 2 months. And then when you're out from your vaccination more than 2 months, you start to see more and more COVID cases to the degree that you are maybe 8 times higher than if you have hybrid immunity, or about 3 times more cases than if you are recovered from a COVID case. So this would suggest that your best condition is to have gotten COVID and then to have gotten the vaccine afterwards, because that's what this group down here is that they got COVID and then they got vaccinated and then they have the absolute best protection. But they only look at it for 4 months, right? So it does rise. The longest that they look at is 6 months.
This is just about infectivity too, right? This is nothing to say nothing of people's symptoms or anything.
Yeah, exactly. But so this was just a very interesting thing. I remember somebody asked about hybrid immunity. And so what it would seem to suggest is that hybrid immunity of any form, vaccine, then COVID, COVID, then vaccine, is better than either COVID alone or just getting the booster. Which I think has to do with epitope updates, because you're like seeing different versions of the virus and then you're reacting to them, as opposed to just seeing this form that was frozen in time when they produced it.
Yeah, there's also like the idea that the vaccines are specifically engineered to not lead to a robust response. Right? Because you don't want people, like, it's very hard for us to keep in mind that the big killer of this disease is your own immune system. It's really unfortunate piece of this.
Yeah. So looking more at the Israel data, they show there wasn't an additive response to the second dose, further cementing their point that like maybe it's going to be okay. Then they're like, most people already have it. Like Dr. DeLay was saying, you have a seroprevalence of 80% in the Central African Republic. You have...
They make the claim that we might be underestimating our seroprevalence in Europe and North America by 10-fold, maybe as much as 50-fold in Southeast Asia, which is pretty wild if you think about it. I mean, they're basically saying everybody's seen this.
And so if everybody's seen this and we look at the data coming out of Israel, do we have the same necessity for a second shot? And the data from Israel would seem to suggest that a single shot is okay. I will say that the data from Israel is not broken out by age. It's not broken out by any kind of health state. And so it's one of those cases where you see a broad statistic and you can't make full conclusions off of it. You would have to look deeper to see how it breaks down according to individual experiences.
It's also pretty wild to ask these, or you know, the ask is pretty intense to people to get a medical treatment for something that, you know, in a lot of these people's cases, they're asymptomatic, they're seroprevalent. They get tests, you know, if they're seroprevalent, but they don't even recall having been sick, and now you're asking them to take a medicine, and they're like, "Hey, I've never been sick." That's a hard ask for people,
especially in places where trust in medicine is less than it is here. Perhaps Africa has a long history of being at the butt end of medical experimentation, and so I think that there's a very real and genuine distrust of these systems and these organizations that on one hand are helping, but on the other hand are maybe not really making things better. To kind of close it down, they basically go through a list of things that make you likely to get really sick, right? And so they're basically like 75% of all admissions and more than 90% of deaths happened to those above 70, compared to 0.4% of deaths and 7% of admissions in those below 50. So they're building their argument. They're suggesting that, hey, there's people who this is really dangerous for. People who have Down syndrome, severely immunosuppressed people who have very frail health. These people definitely warrant increased protection. People who are old warrant it. But
there's sort of there's more than one reason to shunt the supply towards these people who need it too, because you know distributing a lot of medicine to people who aren't going to benefit from it hasn't worked out in the past, and we won't really know what's happening for another 50 years. This was a disaster for us with antibiotics. I can still go to the grocery right now and find antibiotics in everything, which is crazy to me. It just makes me want to scream. But antibiotics are starting to not work. This is a real problem in hospitals all around the world right now. And that's because the bacteria have evolved around them. And we're going to have to watch out for that because antiviral resistance, viral evolution is something we're just starting to study. We don't know anything about it. Well, we know very little. And so hastily treating people that don't need the treatment is potentially an open back door.
And especially because you can really look at the data and you can show that there are people who greatly benefit from a second dose, and they should be the ones that are getting it. And you can make statistical predictions as to who those people will be based off of their medical characteristics. And they show here that in these vulnerable populations, after the second dose, you get a huge reduction in likelihood of death, 83% reduction. And all of the remaining deaths were occurring in the 5% that were the absolute highest risk category, which means that if you can really focus your treatments, you can create a condition where you are able to genuinely help the world by distributing one dose of vaccine and then also really focusing on using the extra vaccine to treat people that need it the most. And again, I mean, the rest of it is basically just an argument for let's be smart about this. We have all these technologies, we have all of these things that we've done, but let's not go crazy. Let's give them to the people who need them, as opposed to treating them like this mandate for every single person on Earth.
I want to add one more thing. We're trying really hard not to bias you about all of this. These are actually the two most cited papers on Google Scholar, by the way, concerning vaccines at all.
Yeah, we're always trying to find the papers that are.
So I didn't just like cherry pick these necessarily. I mean, I decided to pick the most referenced papers, but they are the most cited papers. So, the conclusions seem a little radical, but they're actually quite standard. What would you call it? Center stage material in the scientific community.
Which is a great example of how the discourse about what science says in the news and in culture does not reflect what is written in these kind of boring and dry papers that you have to really knuckle down and get through. And like always, it's easiest to understand the world not by trying to filter the secondary information you get, but it's to just go and look at the community. What are the scientists saying? What are they talking about in their papers?
This is such a great paper, by the way. This comes from everywhere. This is written by so many different scientists from so many different countries. They're all working together. This is awesome. They're from everywhere from Oxford to Bangkok, MD Anderson.
What's MD Anderson?
Or sorry, Maryland. MD Anderson's a hospital.
New
South
Zealand,
Africa. Like this is
India,
people all around the world.
Mozambique.
These are scientists all getting together and contributing to this paper. This is an awesome piece of work.
Yeah, I hadn't even noticed that. That is really fantastic.
So we actually only have like 10 minutes left for questions, but I think we should do them.
We got 15. Let's do it.